We are told that COVID mRNA vaccines aren't gene therapies, and that mRNA vaccine does not change human DNA. What is the truth about these claims? To find out, see our YouTube channel at RealView Report and subscribe for more interesting videos. The mode of action of COVID-19 mRNA vaccines should classify them as gene therapy products, GTPs. The Alliance for Science is a global communications initiative to promote an enabling environment for science-based solutions to challenges of food and nutrition security, climate change, global health, and misinformation. It promotes genetically modified organisms, GMOs, based agriculture, and has Bill and Melinda Gates Foundation as its primary source of support, main funder. According to it, some COVID vaccines use genetic engineering, and the mRNA-based vaccine by Pfizer does not change your DNA as mRNA stays in the cytoplasm, where the ribosomes are. It does not enter the nucleus and cannot interact with your DNA or cause any changes to the genome. Thus, an institution which promoted GMOs, which is based on genetic engineering and DNA modification, says that genetically engineered COVID vaccines do not change DNA. Let's find out whether Pfizer's COVID vaccine can change your DNA or not. On the 7th of June 2021, AGC Biologics announced its partnership with BioNTech Shade to supply plasmid DNA, pDNA, for its Pfizer-BioNTech COVID-19 vaccine. According to its website, our expertise includes experience with E. coli, Pichia, Pseudomonas fluorescens, Bacillus microbial expression systems, and more. The authors of this New York Times article visited Pfizer's facility in Chesterfield, Missouri, where trillions of bacteria are producing tiny loops of DNA containing coronavirus genes, the raw material for the Pfizer-BioNTech vaccine. A plasmid is a small, extrachromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. They are most commonly found as small circular, double-stranded DNA molecules in bacteria. In Pfizer's vaccine, a coronavirus gene is added to plasmid DNA of E. coli bacteria. The coronavirus gene contains the genetic instructions for a human cell to build coronavirus proteins and trigger an immune response to the virus. Later, after other processes, the DNA containing the coronavirus gene, or the spike protein of only the original SARS-CoV-2, is converted into an RNA molecule through a process called transcription. Transcription is the process in which a gene's DNA sequence is copied, transcribed, to make an RNA molecule. Messenger RNA, mRNA, is a subtype of RNA. An mRNA molecule carries a portion of the DNA code to other parts of the cell for processing. This mRNA is then put inside lipid nanoparticles, LNP, in order to protect and help the mRNA to enter human cells. According to Bill Gates's GAVI, some viruses like HIV can integrate the genetic material into the DNA of their hosts, but this isn't true of all viruses, and HIV can only do so with the help of specialized enzymes that it carries with it. mRNA vaccines don't carry these enzymes, so there is no risk of the genetic material they contain altering our DNA. However, mRNA isn't the same as DNA, and it can't combine with our DNA to change our genetic code. It is also relatively fragile, and will only hang around inside a cell for about 72 hours, before being degraded. So, the two important questions here are: 1. Can the mRNA enter the nucleus, which contains the human DNA? 2. Can the mRNA integrate itself with human DNA and change it? In the 5.3.6 cumulative analysis of post-authorization adverse event reports of PF 07302048, BNT 162b2, received through the 28th of February 2021, the first adverse events of special interest which is not caused by the Pfizer vaccine is 1p36 deletion syndrome. 1p36 deletion syndrome is a chromosome disorder. A chromosome disorder is a change in chromosome number or structure which results in a set of features or symptoms. People with 1p36 deletion syndrome have lost a small but variable amount of genetic material from one of their 46 chromosomes. A chromosome is a thread-like structure made up of DNA found in the nucleus of each cell, which carries hereditary information in the form of genes. And among the major characteristics of the syndrome are craniofacial dysmorphism, structural brain abnormalities, seizure disorder, hearing loss, intellectual disability, and growth delay. Deletion of 1p36 is a frequent segmental chromosomal loss found in different human cancers, including those of neural, epithelial, and hematopoietic origin. This means that the loss of genetic material from the short arm of chromosome 1 at position 36. Chromosomal deletions can lead to the loss of important genes, potentially contributing to the development of cancer. So, the first adverse event shows that the mRNA can enter the cell nucleus, otherwise how can it delete a part of chromosome? This leads to the second sub-question. How did the chromosome 1p36 get deleted? CRISPR-Cas9, CRISPR-associated protein 9, is a revolutionary gene editing technology. Using this technology, DNA is cut up with incredible precision so that parts of the DNA can be removed, for example, to correct unwanted mutations, or other parts of DNA inserted. Essentially, CRISPR allows scientists to edit the building blocks of life, much in the same way that computer programmers add or remove HTML code. It can be used as a tool to create genetically modified organisms or fix disease-causing mutations in people. To put it simply, CRISPR-Cas9 acts as a pair of molecular scissors. It cuts DNA at specific locations and either deletes sections or replaces them with alternate sequences. CRISPR-based systems use a designable guide RNA that directs a bacterial-derived gene editing protein, such as a nuclease, a base editor, a transposase, recombinase, or a prime editor, to the desired genomic target. While the Pfizer vaccine uses mRNA and lipids or lipid nanoparticles, LNP, it doesn't list gRNA among its ingredients. However, mRNA encoding CRISPR-Cas9 can provide an effective and convenient approach for cell-selective genome editing and targeted gene therapy. So, this is how they deleted the chromosome 1p36. It is also interesting to find that Pfizer announced a collaboration with MIT's Synthetic Biology Center, MIT-SBC, in February 2014. In April 2014, the United States Patent and Trademark Office granted patent to Broad Institute, MIT, and Dr. Feng Zhang. Zhang invented a way to use CRISPR to edit the genomes of mouse and human cells. And to use the technology, permission of Zhang is also required. And interestingly, Zhang is also a professor at the MIT-SBC, which had a partnership with Pfizer. The second question: can the mRNA integrate itself with human DNA and change it? We had seen earlier that some viruses like HIV can integrate their genetic material into the DNA of their hosts, but this isn't true of all viruses. And coronaviruses were not known for integrating the genetic material into the host genome as part of the natural life cycle. However, SARS-CoV-2 RNAs can be reverse transcribed and integrated into the DNA of human cells in culture, and that transcription of the integrated sequences might account for some of the positive PCR tests seen in patients. SARS-CoV-2 RNA can be reverse transcribed and integrated into the genome of the infected cell and be expressed as chimeric transcripts fusing viral with cellular sequences. Reverse transcription is the synthesis of DNA from an RNA template. This process is driven by RNA-dependent DNA polymerases, also known as reverse transcriptases. What this means: your DNA has been edited like in genetically modified organisms, GMOs. When a foreign DNA is introduced into an organism, it is called a genetically modified organism or transgenic organism. This makes a whole new subcategory of GMOs called genetically modified humans, where a foreign or alien DNA has been introduced in your body. However, if foreign DNA is introduced into the body, e.g., through infection with a virus or exposure to external sources, the immune system may mount a response to eliminate the foreign material. This response may involve the production of antibodies against specific components of the foreign DNA or the activation of other immune cells. Interestingly, double-stranded DNA antibody positive is a side effect of not taking the Pfizer vaccine. The term double-stranded DNA, dsDNA, antibody positive, is often used in the context of autoimmune diseases, particularly systemic lupus erythematosus, SLE. It indicates the presence of antibodies in the blood that specifically target double-stranded DNA. Autoimmune disease systemic lupus erythematosus is another side effect. Here, the body's immune system attacks your own tissues and organs, autoimmune disease, and cause inflammation of multiple organs or organ systems in the body. From these, it can be confirmed that our DNA has been contaminated, causing our immune system to attack foreign or alien components of DNA, or the DNA itself. Thus, our DNA, which is the invaluable inheritance passed down through many generations, and which can be traced back to the first human beings on Earth, carrying the instructions necessary for their growth, development, functioning, and reproduction, is now contaminated or perverted. We have heard about corporate colonization, which refers to domination by corporate organizations by invading and taking over other countries. Now, they have extended the frontiers of their colonization by invading the core of human beings and changing their DNA. Or, in other words, corporations have literally colonized not just our very bodies but our future generations as well, who will continue to carry this alien DNA. Practically speaking, corporations have patented human beings and have started to rule over our bodies. If you do not own your body, you own nothing. But Bill Gates is not interested in bodily autonomy, or people exercising choice with respect to their bodies, in case they become more healthy. And by making genetically modified humans without even informing us, these corporations and the scientists working for them treated us as lab rats, and turned the whole world into an open laboratory for their nonsensical experiments. All this, while asking us to trust the science. It is important that those who have taken mRNA vaccines should have their DNA checked, to find out whether mutations have indeed occurred. And, now is the right time for genetically modified humans to take vaccine selfies or vaxies, or do a vaccine happy dance to celebrate this grand event in the history of humanity.